Hello everyone. This section is for endospores, sporulation, and germination. After learning this section, you should be able to describe in general terms the process of sporulation. Describe the 3 stages that transform an endospore into an active vegetative cell. Sporulation is the process of making endospores. Endospore-forming bacteria are common in soil, where they must be able to withstand fluctuating levels of nutrients. Sporulation normally commences when growth ceases due to lack of nutrients. Thus, it is a survival mechanism that allows the bacteria to produce a dormant cell that can survive until nutrients are again available and vegetative growth can resume. These bacteria cycle between 2 states: vegetative growth and survival as an endospore. Vegetative growth is a normal, continuous cycle of growth and division. By contrast, sporulation is a complex process that occurs in a highly organized fashion over several hours. Look at the picture. It shows examples of endospore location and size. The mature endospore is located in a characteristic location in the mother cell, referred to as the sporangium. Depending on the species of bacteria, endospores may be centrally located, close to one end. We also call it subterminal or terminal. Sometimes an endospore is so large that it swallows the sporangium. We call it swollen sporangium. One of the best studied endospore former is Bacillus subtilis, which is an important model organism. Sporulation may be divided into 7 stages, as seen in the following picture. Firstly, the cell's DNA is replicated. This is stage 1, followed by an inward folding of the cell membrane to enclose part of the DNA. Then, the phosphor spectrum is produced. This is stage 2. The mother cell membrane continues to grow and engulfs the immature endospore in a second membrane. This is stage 3. Next, cortex is laid down in the space between the 2 membranes, and both calcium and dipicolonic acid are cumulated. This is stage 4. Protein coats are formed around the cortex. This is stage 5, and maturation of the endospore occurs. This is stage 6. Finally, lytic enzymes destroy the sporangium, releasing the spore. This is stage 7. Sporulation requires about 10 hours in Bacillus megatherium. After learning the formation of endospores, let's see how does the endospore germinate to vegetative cell. The transformation of dormant spores into active vegetative cells is almost as complex as sporulation. It occurs in 3 stages. The first is activation. The second is germination. And the third is outgrowth. Activation is a process that prepares spores for germination and can result from treatment such as heating. This is followed by germination, the breaking of the spore's dormant state. This picture, we can see the cell of Clostridium pachydermum emerging from the spore during germination. Germination begins when proteins called germinant receptors located in the inner membrane detect small molecules such as sugars and amino acids. Upon detection of these molecules, a series of events occurs, including release of the calcium DPA complexes, breakdown the peptidoglycan in the cortex, and water uptake. Eventually, water levels inside the germinating spore reach those characteristic of vegetative cells, and enzymes in the core become active. This allows the spore to begin synthesizing the various molecules it needs to initiate spore outgrowth and return to a vegetative state. Okay, that's all for this section. Here is a word list for you.